All right, let's do an example problem from chapter 6. Okay, so this problem is mercury levels in tuna. We'll just read it quick. Rising mercury levels due to industrial pollution is a recent concern. Many fish have high levels of mercury. Data were collected on random samples of tuna from 1991 to 2010 and are available in the file tuna. Each row in the data set represents the mercury level of a different fish. Do fresh yellowfin tuna have different levels of mercury on average compared to fresh albacore tuna? Use the fresh tuna data to answer the following questions. Okay, so it sounds like we've got a sample of two different kinds of tuna, and we want to know about the levels of mercury. That sounds like probably a numerical variable, so it looks like we've got a two mean problem coming up, and let's work our way through the problems. So is this an experiment or an observational study? Explain, identify the observational units, identify the explanatory and response variable, and identify whether it's quantitative or categorical, and then define in words the parameters of interest for this study and assign symbols. Okay? So it's an observational study. The groups were already in existence, albacore and yellowfin tuna, they already exist, and we just observe the mercury levels in each sample. The observational units are the 231 yellowfin tuna and the 43 albacore tuna that were caught between 1991 and 2010. It said it was a random sample, so hopefully these are representative of the populations of yellowfin tuna and albacore tuna. The explanatory variable is the type of tuna. It's either yellowfin or albacore. That sounds like a categorical variable. It's this or that, yes or no, that kind of thing. The response variable is the level of mercury. I'm assuming this is a quantitative variable, some number of grams of mercury per kilogram of fish or something like that. Probably milligrams or something like that. All right, and then the parameters of interest. So what are we studying? Well, we want to know what is the true mean level of mercury in yellowfin tuna and the true mean level of mercury in albacore tuna. And the symbols are mu y and mu a respectively. These are parameters, right? These are what is true in the whole population. So it's a mean. It's the mean level for all the yellowfin tuna and all the albacore tuna. All right, and then number E says state the null and alternative hypothesis in words as well as symbols, and that would be this right here. So the null hypothesis is that the mean levels of mercury are the same in yellowfin and albacore tuna. The alternative is that they're different. So in symbols, we can say mu y minus mu a equals 0. This is another way of saying that they're the same if the difference is 0. And if the difference is not 0, that's another way of saying that they're different. So we're saying that the average level of mercury for yellowfin tuna minus the average yellowfin, or the average level of mercury for albacore tuna, if you take that difference, you get 0. That's our null hypothesis. We assume the two kinds of fish are the same, but maybe they're different. Maybe the type of fish matters. Okay, let's keep going. So if we're gonna do a statistical study, what is our statistic we're gonna use? We'll take that one next. So the statistic should be the observed difference in the sample means. We don't know, right? We don't know what these population means are, but we could take a sample, and we did, and then we could subtract the two. So there's going to be an X bar for the yellowfin tuna, there's going to be an X bar for the albacore tuna, and if we take the difference, we get a number. In this case, it's not 0. And where does this number come from? Well, it comes from the applet, so that's the next thing to do. So let's see what is the data going to look like in the applet. So that's what they say. Use an appropriate applet to carry out the analysis. All right, so the data is down here under chapter 6 under tuna. So if we here's all the data. So it says the kind of tuna and the mercury level, and then the kind of tuna and the mercury level is a whole bunch of them. So let's highlight all these up and copy them. And then we'll go to, I think it said simulation based, so we want to do probably multiple means. Let's open multiple means, and we'll clear this out, and we'll paste it in. All right, so this is going to be all the data that we just had, and we will click use data, and that should make for us a dot plot of all the yellowfin tuna data based on mercury level and all the albacore tuna data based on mercury level. If you click this box, you can get a nice box plot. I would say we've got some skew right here for the yellowfin tuna, which is interesting. the albicore is fairly symmetric. And the reason I'm noting that is if we were going to use a theory-based test, remember, what are the conditions for using a theory-based test? Well, the sample size has to be big, bigger than 20, and we've got 43 and 231. So the sample size seems pretty good, but I am seeing some skew in this one. So I would want to note that if I was going to use a theory-based test, I'd be like, well, it doesn't quite fit the conditions because I've got skew in my data set. So instead, let's use a simulation-based test. So that would be clicking this to do a simulation, and we usually do like 1,000 shuffles to get a good thing. What's our statistic? Well, the mean for the yellowfin is 0.35. The mean for the albicore is 0.36. So we could subtract these two, and that would give us the difference. And there must be some decimals hiding because the computer says that the actual difference is 0.003. And I think in the way I wrote up my, I did the yellowfin minus the albicore, so I think I got a negative difference instead of a positive difference. It doesn't really matter as long as you're consistent. Okay, so that's where the negative 0.003 comes from. So what does shuffling mean? That means take all of these numbers, right? This whole list of numbers. I know that this number is from an albicore fish. I know that this one here is from a yellowfin tuna fish, right? But we could just take all these numbers and randomly deal them into two piles and then see if we did it by, say, random chance, what, and then randomly reassign the numbers to say, oh, this was the albicore pile and this was the yellowfin pile. Then what kind of a difference do you get? Well, you're going to get an X bar here and an X bar here, and there'll be a difference, and we can graph that difference make a graph of it, right? And if we do it 999 more times, then we'll get 1,000 randomized differences, and this is the null distribution. This is what you would expect to happen if, in fact, yellowfin tuna and albicore tuna have the same level of mercury, right? If there's really no difference, that's why it's the null distribution. It goes with the null hypothesis. If there really is no difference between the two types of tuna, then this is what kind of differences in our X bars we would expect to get by random chance. And you'll notice the numbers go anywhere from 0 to 0.12. And what was our observed difference? Negative 0.003. That's what we saw in the sample. So where is negative 0.003? Well, this is 0.06. So 0.003 is pretty small. It's got to be somewhere in here. Two-sided. So let's try negative 0.003. Let's do the count. Oh, sure enough. Yeah, that's really close to 0. And so it turns out that 928 out of 1,000 of the random observed differences from the simulation are as extreme or more extreme as what we saw in our sample. And so this is a very typical kind of difference that could happen if, in fact, the two kinds of tuna have the same level of mercury in them. So we do not have very strong evidence at all that there's any difference. But if our observed difference had been something like, say, 0.1, maybe, then we could have drawn the line at 0.1, and then you see, oh, okay, only 2 out of 1,000, right? So in our case, the observed difference was right in the middle of the distribution, so we didn't have strong evidence. If it was, a bigger number, then you might have had strong evidence, but we did not have very strong evidence. So what's the p-value? 0.928? Okay. So yeah, I think the last time I did it, I got 0.936. When you do a simulation, it's going to change slightly each time, the p-value. So running the simulation generated a large p-value, so we did not have evidence to conclude the average mercury levels are different for the two types of tuna. The observed difference could very plausibly occur even if the population means were the same. And then question H says, make a confidence interval. All right, so we can do that. We can make a confidence interval. Our formula is our statistic, which is our observed difference, plus or minus 2 times the standard deviation of the null distribution. If you go back, what is the standard deviation? 0.0035. I think it was 0.0036 the last time I ran it. So if you double that, you get 0.072. And if you add and subtract that to negative 0.003, you get these two numbers, and that's my confidence interval. So the interpretation is we are 95% confident that the true difference in mercury levels between yellowfin tuna and albacore tuna is between these two numbers. And I like to say which way I subtracted it, yellowfin minus albacore. And that's important, especially if this doesn't include 0, then what you're saying, if this is, say, two positive numbers, then we're saying that the yellowfin would be bigger than the albacore. But in this case, we don't really have evidence that one is bigger than the other. I mean, it could be that y is bigger than a, it could be that a is bigger than y, we don't know. But 0 is included in the interval. All right. And then they want to say, give a full conclusion. Talk about significance, estimation, causation, generalization. Kind of wrap all this whole problem up in a bow. So we've already done significance and estimation, causation and generalization. We'll kind of grab here in a second. So because our p-value was large, it was so large, we don't have evidence that the true population mean levels of mercury are any different between the two kinds of tuna. It's plausible. They could be, but we don't have evidence that they are. Our simulation-based confidence interval gave us that. So we're 95% confidence that the true difference between the true average mercury level in yellowfin tuna minus the true average mercury level in albacore tuna is between negative 0.075 and 0.069. 0 is in the interval. It's plausible the two means are equal, and it's also plausible there's a difference somewhere, anywhere in between those two numbers could be. Causation, if we had found an association, we did not, but if we had, we would have been able to say that it was different in the two types of tuna, but not necessarily why or how, right? We're not doing a randomized experiment with cause and effect. We didn't assign treatments. We're just sort of saying that this population has a different level than this population, and not like why or whether it was a causing effect or whatever. And then the generalization. The study involved two types of tuna. The samples were random samples from those two species. So the conclusion should generalize to those species in which the samples represent, but probably not to all fish. And I don't know if they were only, Pacific tuna, maybe then it would generalize to Pacific tuna, or if they were sort of representative tuna from all over all the oceans or something like that. But that's the kind of thing you could say with generalization. So we do have random samples where, most likely these are representative of the populations from which they came. The last thing I want to do just briefly is what would have this looked like if we had done theory-based inference instead. So if we'd done theory-based inference, we would want to use the 2-mean applet, and we could either have pasted in the sample mean, sample standard deviation, and sample size. And we could have done that. Those data are given here, the sample size, sample mean, sample standard deviation. So it could be that you're just given that data. But in this case, we actually had the data, so it's probably just as easy to clear this out and paste it in. Oh, we must need this one because they're all stacked up on top of each other. Okay, then we got the two groups. Right. Okay, and this time again, this difference is so small that it's not even displaying it. But then if you click, test of significance, the theory-based applet is going to give us, a similar picture. It's just going to draw the null distribution with the standard deviation that it's calculating from the formula instead of simulating, and you don't need to know that formula, but that's what's going on. And you can see the p-value is very large. It's not exactly the same as what we got, but pretty similar. The t-statistic is just saying, we're not even one standard deviation away from the mean. And then it will cook out a confidence interval for you, which is kind of nice. It's not exactly the same as what we got, but it's kind of similar. And a theory-based and a simulation-based are going to be slightly different. Okay, so that's what it would look like in a in the theory-based applet. And then you probably just want to note that because you've got some pretty good skew in the yellowfin tuna, that the validity conditions are not met in this data set. So you really, if the problem said use a theory-based test, you should probably say, well, this skew is here, and that's not good. You don't want that to use the theory-based test. And that's probably one of the reasons why we're getting, some difference between our standard deviation here and the standard deviation and the null distribution here, right? They're not quite the same size, and usually they're closer. All right, so that's how you can do a problem from chapter 6 where you've got two means, and those are kind of typical questions that you might expect to see.